Okay, welcome. This is Issues in Biotechnology, otherwise called The Way We Work with Life. It's a general education course that satisfies the natural sciences requirement at the University of Rhode Island, and it's meant as a sweeping general survey of life and biotechnology. And I think what we will see is that biotechnology these days challenges a lot of our thinking about life and how we work with it. Nearly every aspect of biotechnology as it's applied today is somewhat controversial for one reason or another. So I think what you'll see during this series is an eye-opening experience of what is happening now in the biological sciences and how those sciences are applied. And I think what it will also point out are the opportunities for the future. So you can use this as a lens to look into the future based on what is happening now in the biological sciences. So in the first part of this class, we explore the question, what is life? From a very basic level. From its information content level, we understand life as an information processing system that is capable of replication with variation. That's a very sweeping statement, but it covers pretty much all of the diversity of life as we understand it on this planet. And I don't want to go back and redo the first half of this class, but we ask this question, what is life, in the first part of this course, laying the groundwork for then how are we now capable of working with biological life in the context of biotechnology. So in the first part of this class, we're interested in how we go from the information that is stored in DNA out to an organism that's capable of discussing it. How do we go from the information in DNA to its expression through RNA and eventually to proteins which cause traits? And then how are we capable of manipulating DNA now in the context of modern biotechnology? So what is the biology of life? Once I think we have an understanding of life as biology, I think this puts it into context, and hopefully you will never view an oak tree the same, or the grass that you walk on, or your hand, or the foods that we eat. And it also brings up a number of other interesting questions. Why is there duality? Certainly there is a common occurrence of duality within biology. A right and a left, an up and a down, a dorsiventral, a shoot and a root. These are common situations that we observe throughout biological life. There are exceptions, of course. There are radially symmetrical organisms, starfish and sea urchins, but predominantly we see this bilateral symmetry and we see polarity. This should cause us some reflection philosophically, that while we see this duality throughout biological life, we ourselves as humans understand things in terms of right and wrong. Why is that? Is that a further reflection of our own biology? So the first part of this class, what is life, we get to look at the biological situation, but also how this reflects on certain philosophical concepts. What is biotechnology? Well, biotechnology is taking anything that is in life and using it for use by humans. And humans have actually been in biotechnology for a very long time, and I'll explain that during the first part of tonight's lecture. Biotechnology today, though, as a word, commonly refers to DNA-based biotechnologies. While humans have been involved with manipulating plants and animals for their use for thousands of years, using DNA and manipulating DNA in modern genetics is a relatively new enterprise. So how do we work with the stuff of life? This involves aspects of modern biotechnology, including how we sequence DNA, how do we clone genes, how do we move genes from one organism into another. So during this part of the course, I want to enter into how we work with life through the explanation of some of these techniques and how they apply to agriculture in terms of modern advanced breeding technologies as well as genetic modification, how they apply to pharmaceutical biotechnology, including vaccine development, small molecular drug designs, pharmacogenomics, personalized medicine, and so on. Medical biotechnology, including tissue engineering, stem cells, cyber interfaces and bionics and nanotechnologies. We will look at the biology of cancer and emergent technologies that are related to DNA and understanding cancer through that context. We will also look at how biotechnology will influence how we might encounter the next pandemic. And I say next as this is inevitable. And we will look at the pandemic flus that have occurred in the past and how this helps us understand what will happen in the future and how we might be best prepared for that. And then along with that, we will see that there is a dual use to that knowledge, that the darker side of understanding pandemic flus may in fact influence that same knowledge to be used in terms of bioweapons. We will also examine how DNA is currently used in forensic analysis and the controversies that that creates about DNA databasing in the public in general. So those are some of the topics that we're going to cover in the next few weeks on applications in biotechnology, and that's a lot in itself, but really that's only the surface of how this influences our life these days. So the second half is on the applications of biotechnology. So the first part, we will consider agricultural biotechnology. And as I said, humans have been using plants and animals for thousands of years. So in the first part of considering agricultural biotechnology, I think it's helpful to get a perspective on where our food comes from. So where does our food come from? Sometimes I'll ask students this and they'll say, it comes from the grocery store. Well, yeah, that's an obvious first answer, but really, where does our food come from? Not only how is it grown on a large scale, agriculturally, how does it come to the grocery store, but even before that, what are the genetics? How did we select out of wild plants and domesticate these wild plants in order to become useful to humans? And then next after that, what are the tools that come to bear on modern biotechnology? So that part two will be addressing DNA-based biotechnology and modern agriculture. And that includes genomics, sequencing all of the genes in major crops, advanced breeding technologies as they use that type of information, and then genetic modification. But this also brings up many questions. How do we work with life? What are the tools? What are the ethics that this impose upon this ability? Should we be doing this? What are the controversies? If we say that biotechnology is controversial, why are these topics controversial? So what? We work with life. Why does this cause so many questions? Why so many mythologies? Why are some people opposed to genetic modification of plants and animals? What are the facts? What are the opinions? What do we think about them? How do we feel about them? Do we believe that nature knows best? That we should just leave it to nature? Or should we get involved? Does human nature know best? When should we intervene? Should we intervene at all? Should we pursue genetic improvement of other organisms for our benefit as a society? Should we be manipulating plants? Or perhaps we were better off hunting and gathering? Can we use biotechnology to feed the poor? Realize that now the planet is at 7 billion people. During your generation, that number will go to 9 billion people, with resources becoming more and more limiting. Energy resources, water resources, land, arable land especially. Soil conservation becomes critical. Out of 7 billion people now, half of those live on rice. Half of those live on less than 2 cups of rice a day. During the next increase in world population, most of that will happen in developing countries. Who will feed those people? I asked this question once, can we use biotechnology to feed the poor? And a student asked me, should we use biotechnology to feed the poor? And I said, really? What do you mean by that? And he said, well, if we feed the poor, they will just make more poor. I'm like, really? Whose decision is that? Yours? So we can see that our values depend on perhaps our own situation and how this may apply globally. Should we be manipulating animals? We domesticated plants thousands of years ago and domesticated animals too. Now we have the capability of genetically modifying plants with introduced DNA, and I'll get into that in the second part of this lecture tonight. But we now have that capability of doing this with animals as well. And we have the ability to clone animals. We've had the ability to clone plants for a long time, and we can understand that too. And why do we think of these things as different? So, agricultural biotechnology is the topic right for now. Where does our food come from? Are there any foods or ingredients that you have avoided or eaten less of lately? When you ask this question to the public now, about half of people in our population will say yes. That might include fats, it might include sugars, it might include various different ingredients. It might include GMOs in plants. But about half of people right now, according to an IFIC poll, would answer yes to that question. So where does our food come from? Let's look first at a history of agriculture, and then at plant domestication. One thing that we must say is that in the origin of agriculture, that agriculture itself presents itself as a crucial event in human history. If you were to ask what are the top several inventions by humans, fire might fall at the top of that list. I'm not sure, but fire was a good day. After that, there were several other really interesting improvements. The wheel was also good. not accomplished by all societies. Native Americans, for example, did not have a wheel. But agriculture. Agriculture occurred in many different centers around the world, roughly starting about 10,000 years ago. Before that, humans were hunting and gathering as migrant tribes. We can say that as a crucial event in human history, it was the beginning of civilization. Moving out of hunting and gathering allowed for the accumulation of surplus food supplies. Being able to have surplus food supplies allowed for the other events of civilization, including writing, property, law, property law, and other aspects that came with Neolithic developments. We did not have writing until we had agriculture. So the passing down of information from one generation to the next through writing was only because of a reliable surplus food supply that allowed people to settle down in one place for long enough to have these other events occur and to interact with neighboring peoples. So agriculture was in fact a crucial event in human history, perhaps right up there with fire. And if you look at the cradles of agriculture, this occurred nearly simultaneously around the planet. And we could wonder why about that, and a lot of people have thought about that anthropologically. But certainly one of the most notable areas is that area that we refer to as the Fertile Crescent, where the Tigris and Euphrates rivers join in the Middle East, in Iraq, Syria, and where everything is now embroiled. But that area gave rise to the domestication of lentils about 9,500 years ago. Wheat, flax, rye, all came out of that area at around that same time. Goats were also domesticated at around that time as one of the first domesticated animals. In Mesoamerica, we had the development of squash, corn, zea mays, potatoes from Peru, and beans. Central America, we had cassava and maize developing in the area around Oaxaca, Mexico, roughly around 9,600 years ago. And that's been heavily studied, the domestication of maize. Rice came out of two different species, nearly in parallel, one from China and one from Africa. But the domestication of crops together all accumulated to the ability to have a surplus of food supply that could be depended on from one year to the next. This book by Jared Diamond, Guns, Germs, and Steel, examines the question of the fates of human societies, which is very interesting. Why did some societies succeed and other ones did not? And this is an interesting question that is not easily answered, and it has a lot to do with various factors, one of which would have been agriculture and the development of agriculture, along with other innovations, hence the title. But we now know that humans originated in Africa. In this form, humans had an African origin. We know this from various lines of evidence at this point, both anthropological, but most intensely now at the genome level. We also know the admixture of other hominids that contributed to our modern genetics, including Neanderthals. The Neanderthal genome has been sequenced, and we can now line that up with the human genome and see its contribution to various human genetics, as well as other admixtures that occurred in different locations. But we can see that out of Africa, humans moved into these various locations. And most evidence now supports that humans entered North America roughly 14,000 years ago, probably over the Bering Strait, and moved very rapidly down North American coast into South America to Tierra del Fuego in only a few thousand years, bringing with them dogs. The New World had no dogs. So also, centers of agriculture are pointed out in Guns, Germs, and Steel as providing this basis for civilization. And we can see that in some of these provided a leg up for some of those budding civilizations, some of which who had agriculture rose quickly, developed civilization, and then subsided due to other factors. So where do the plants that we eat come from? Well, one thing that we can notice right off is that nearly all the plants that you have now in the grocery store do not grow in the wild. All of these plants, when you walk into a grocery store, all of these plants that you see would not exist without human intervention. They didn't exist before humans, and likely if humans disappear, so would these cultivars. So all of these plants are dependent upon humans and would not exist if humans had not intervened in the first place. So you could ask the question, are all of these plants natural? They wouldn't exist without humans. They require humans for their reproduction. So all cultivated plants then are the result of human intervention, and we could say even the result of human invention. So these plants would not exist without humans. So the importance then of wild plants to plant domestication is one thing that we can consider right off. All of these plants had wild precursors, and at some point, some human had to say, hey, look, that one has some interesting traits. We're going to try to grow that. And from its offspring, kept selecting and selecting for traits that were useful to people. So it was through this process of observation and selection that we derive the varieties that we have today. All of these different varieties. So I want to talk first about the importance of plant breeding and selection out of the wild. Fair enough? So let's talk about plant breeding. So you might ask, if I'm going to talk about plant breeding, why am I showing you a picture of 3 golden retriever puppies? Well, because of the obvious importance to the selection process. All dogs are derived from wolves. And if you look at these puppies, what were they selected for? Well, they're cute. They're cute. They're cute golden retriever puppies. And besides that, they're friendly to humans. You know, and they're relatively bright, as dogs are selected for, but they didn't exist in the wild, and they wouldn't have existed without human intervention. So the variation in dog breeds that we see are a direct consequence of selection by humans. There are over 400 different purebred varieties of dogs. All of these derive from a wolf ancestor. We know this now by looking at the DNA sequence of dogs and comparing it to wolves. This is not open to just subjection. Also, we now know that there is a single gene that's involved in the variation of dog size. If you look at the variation in dog size, it's more than most animals. From Chihuahua out to Great Dane, this is all controlled by a single allele. The IGF-1 allele in dogs is the major determinant of size. And this was published in a recent issue of Science, the journal Science, on the domestication and breeding of dogs. And the Asian wolf is now accepted as the most likely ancestor of all domestic dogs. And that genetically, these can all be traced back to this wild Asian species between 15 and 40,000 years ago, before the first agricultural human societies, in fact. So I think this is a striking example of how humans have selected and bred for characteristics in a complex organism. Another really cute puppy picture as a bulldog. The bulldog was also invented by humans. And if you look at the domestication and breeding of bulldogs, this illustration shows the somewhat shocking deformation of the bulldog head that occurred just between the years of 1890 and 1935. So the compression of the bulldog's face, this shortened face that some people find endearing, also causes the dog breed serious medical problems. So in its selection process, certain deformities also came with those genetics. And that's also true for other dog breeds. Dalmatians, for example, 40% of Dalmatian offspring are deaf. How well would a deaf dog survive in the wild? So a lot of the characteristics that humans have purposefully selected for their companion as man's best friend have in fact made these animals less likely to be able to exist without humans at all. So we've invented these dog breeds and selected for them genetically and maintained them by actively selecting for this process. That brings me to plant breeding and the apple. And the apple is an interesting story all unto itself. The apple of one's eye. What does that mean? Certainly the apple is considered throughout many different stories and mythologies, from William Tell to Eve in the garden. But the apple, where did the apple actually come from? What's the origin of the apple? Well, it turns out that the ancestor of the modern apple comes from an origin somewhere around Kazakhstan. And its wild species, Malus civicei, existed in Kazakhstan, and it's barely edible. Maybe by rodents, it does okay. Small. And it's genetically very diverse. It's an outcrossing species. Coincident with the selection of wild apples by humans in Kazakhstan was the ability that the Chinese discovered for grafting plants, a horticultural technique where the top portion of one plant can be grafted onto a rootstock and bear fruit from the original plant that was selected, not affected by the rootstock itself. So that horticultural technique, developed about 4,000, 5,000 years ago by the Chinese, was adapted to the apple. When a sport of apple from Kazakhstan that was selected for like, oh, that one over there, that's pretty good. We can eat that one, was grafted and cloned, if you will, by travelers going through that area. So the Greeks and the Romans actually had various different apple varieties growing in Greece and in Italy around 500 BC or so. And the stories already started to develop around the certain Dionysian qualities of apples because of its sugar content. Where did early humans get their sugar? There were a few sources of sugar, really. And because of the caloric value of sugar, humans were drawn to sugar. You got a lot of bang for the buck out of that, and apples then were prized. And Pliny the Elder described about 23 different varieties of apples that the Romans then, in the conquest of England, brought with them to England. And these were all cloned by taking vegetative propagules and grafting them onto rootstocks that would support them. So you could then graft out a whole orchard of one variety of each one of these 23 different apple varieties. So these went to England and were well appreciated because the apple could also be used to make cider. You could take the sweet apple juice, press it into apple juice, and lo and behold, that stuff would ferment and everybody was happy. So it was not only the sweetness that propelled the propagation of the apple. So in England then, the apple was propagated as a fruit for its sweetness and for its juice. And the apple made the trip to North America with the colonial invasion of North America. There were no apples in North America prior to the European invasion. And the apples in North America interbred with American crab apples, and so the genetics spread. And the apple spread further into the frontier as people moved from the Northeast west. But the apples that came from England, not all the varieties survived. We have a colder spring here in the Northeast of the US. And so the varieties that would flower early in spring might have been killed off by a quick cold snap in those days. But the apple then spread into the frontier, actually owing to laws that were passed for homesteading that required that farmers that homesteaded would have to stay there for 10, 20 years or more in order to acquire the land. And then as part of that, they had to set up an orchard. So the apple moved westward on that basis. But apples, when they out-pollinate, due to the genetic heterozygosity, return very quickly to a wild type. And you can see these around in the Northeast. While they're quite hardy and squirrels like them, they wouldn't suit today's market very well. Although many of them have very good disease resistance. Then enter a guy named John Chapman. In the early 1800s, John Chapman did a lot for propagating the apple in North America. Who is John Chapman? John Chapman was a naturalist. John Chapman is a historical figure. He lived in the frontier, and he was fond of apples and fond of biology itself. And there's a lot of mythology about this guy. He threw away his shoes because once he stepped on a worm and was remorseful. There's some other interesting stories that go around about John Chapman. He became known as Johnny Appleseed because he would store apple seeds in burlap sacks and paddle down the Ohio River, giving away apple seeds to pretty much anybody who would buy them. And that's how he made his living. But owing to the heterozygosity of apples, all of these don't breed true. So there were other people in those days who were grafting apples and selling apple seedlings. And hence true varieties of apples were then propagated and the selection process continued. So through the early 1800s until today, domestication and selection of new varieties are developed and propagated through the clonal selection of grafting. In 1893, a guy named Jesse Hyatt discovered a sapling growing on his farm in Peru, Iowa. And he would mow this thing down. He would mow it and try to get rid of it. This damn apple tree kept growing back. And one day he just said, well, I'm just going to let it grow then. And when it grew, it bore fruit, and he thought this was a great apple. And it won blue ribbons at state fairs. And he brought the apple eventually to a state fair where it won a ribbon, and it was discovered by the C.M. Stark brothers, who apparently, the story goes, put it in their pocket. And Jesse Hyatt had named the apple the Hawkeye after Iowa, the Hawkeye State. And the Stark brothers, they rebranded it, so as you will, and called it the Red Delicious. So the Red Delicious apple was invented by Jesse Hyatt in 1893. So apparently those pictures that we see of Eve in the garden holding a Red Delicious are misinformed. Unless that was in Peru, Iowa. And I'm misinformed. But these days, you can go to Geneva, New York, where there's a germplasm repository of a large number of the apple varieties that are in existence today. And so we see a large number of varieties of apples even available year-round in your local grocery stores. And I'm sure you have your favorites. I'm from upstate New York originally, and there is nothing like a Macintosh apple before the first frost, right before the first frost. Afterwards, I sort of lose my taste for it, really. And I don't think it stores so well. But before that first frost, when it's nice and crisp, that's a good apple. But what is a good apple? That depends on your own individual taste. What's a good dog breed? Some people like golden retrievers. Some like pit bulls. Now, there are many, many different varieties of apples. As many as there are dogs, maybe more. All of which are clonally propagated through grafting. And these have been selected for different characteristics, including tartness, like the Granny Smith, the Pink Lady, the Braeburn, another one of my favorites, or more sweet, like the Gala apple or the Fuji, less sweet, and again more tart, like the Golden Delicious, and more tart might be more like the Red Delicious. Now, I think actually the Red Delicious was inappropriately named, but that might be my preference. So all these different apples have been selected by these different characteristics chosen by people. And now we even have the capability of growing these in both hemispheres. And so owing to air traffic, we can have Braeburn apples when ours are out of season. So that brings me back to all of these different vegetables. Like the apple, we have selected for different traits that we like in these different vegetables. And it is really no different than that. So all of these things that people go into the store and they say, oh, all natural. Really? What here is all natural? It depends, I guess, on your definition of natural. I'm also a little bit befuzzled about the word whole foods or other designations. Let's take the genus Brassica. So the Brassica is part of the Brassicaceae, or the cruciferous plants. And there's a large number of our vegetables that are derived from that family. It's the mustard family, shown here in this slide as wild mustard. But the Brassicas give us a wide range of different vegetables, including broccoli and cauliflower and the Brussels sprouts and cabbages and turnips. Radishes are also Brassicas. So this one genus alone has given us a wide variety of different vegetables, all that have been selected for out of a wild cruciferous precursor. So if you consider the Brassicas, here in this slide is a picture of what wild cabbage would look like prior to domestication. So Brassica oleracea as a species, a single species, gives us kale, cabbage, broccoli, cauliflower, kailan, Brussels sprouts, and kohlrabi. These are all the same species. These are all just different varieties of the same species. So like the dog, and you have all of these different varieties from Chihuahua out to Great Danes, one species gives us the diversity from broccoli to cauliflower to broccoli. And all of this derived by human selection. And here are some of the other Brassica oleracea, kale, collard greens, Chinese broccoli, and the like, all derived by human selection. And broccoli, a fantastic variety, was developed originally in the northern Mediterranean region. And since the Roman Empire, broccoli had been valued amongst the Italians and brought to England from Antwerp in the mid-1700s by a guy named Peter Scheemakers. And Peter Scheemakers then was responsible for developing and selecting some of the modern varieties of broccoli that we have today. Not Albert Broccoli. Albert Broccoli was the guy that brought us James Bond movies. So the importance of varietal selection, that's what I mean to underscore here. All of these plants, in spite of what people commonly think, have been derived by humans. And I haven't even gotten to DNA yet. I haven't even gotten to modern advanced genetics. This has all been done by human selection prior to advanced biotechnology. I want to also give you a brief story about some of my own research as it interfaces with this area. One day, this always starts off sounding like a joke to me, but it's really not. One day, a guy came into my office, and he said to me, he goes, "So you're a plant biologist?" And I said, "Yeah, this is my background, by the way." And his name was Peter Salou. And he said, "Can you make a garlic that grows like a scallion?" And I thought, well, that's kind of weird. And a lot of people ask me weird questions about plants. You know, this happens to me all the time, really. And I said, well, what did you have in mind? He said, well, I want a garlic plant that you can eat the greens like you would eat a scallion. And I said, well, you know, I'm kind of busy. So I sent him to a postdoc of mine who then was working at the University of Connecticut. And she developed tissue culture of garlic with him. And he came back a year later and $80,000 with little garlic plants in Petri dishes. And he said, see, we did it. And he was all happy with this. And I said, well, you did what? He goes, well, we have little garlic plants. And I said, well, what do you plan on doing with these? And he says, we're going to commercialize them. And I mean, these things were like this big, right? And I said, well, how much did you spend on this? And I said, how much are you going to sell these for? Really? And, you know, his face just sank. And he said, well, can you come up with a process that would make it cheaper and better? And again, I was like kind of busy with other projects, but the American Society for Plant Biology meetings that year happened to have been in Providence, Rhode Island. And when I got done giving my talk on grass genetics, I walked down the hall and there was a symposium on garlic. And who would guess? So I walk into the symposium and all the world's garlic experts were there, all 12 of them. And I learned a lot about garlic. And they connected me and I started making collections of garlic from around the world. That ended up, I selected out of all of these garlics, a garlic that could grow from vegetative propagules called bulbils. And I developed the process of growing these bulbils into a plant that looks like a scallion that you can eat the leaves and the whole thing thereof. Whoa, let's zoom ahead. The Italian scallion out of Rhode Island was born. And that's what the plant looks like. It looks like a scallion, except that it has flat leaves. And I patented it with Peter Salou, and the patent has issued. So green garlic is now a new vegetable. And after that, that patent issued in 2011. And since then, there are 2 other patents. We patented the methods for commercial production and the chemoprotective properties that this type of garlic has, because you can eat it raw. And the active ingredient in garlic is allicin, and that's destroyed by cooking. So aside from telling you this personal story about how I got involved in varietal selection and garlic, it's the importance of selecting plants out of the wild. People often ask me, how did you do this? You know, was this some mad science? No. I simply looked for a garlic that had the traits that we needed to perform this quality. And you can say, well, how can you patent that? So, well, nobody else did it, right? So the patent office said that that was novel, and I agree. So now that company, we made a company called Ophios, and are entertaining the sale of that company to large producers. But this also brings up the importance of varietal selection to plant domestication. If you look at a seed catalog, like this one from Park Seed Company, these commercial varieties of flowers and vegetables that you see in this catalog are all the result of long conventional breeding programs. Somebody out there, some plant breeder, saying this one by that one, and we will make a seed that can be produced. So how many of these varieties could be found in the wild? How many of these cultivated flowers would exist without human intervention, like all of these plants? What self-respecting poinsettia would be seen in the wild looking like that? These new varieties of fruits and vegetables. How long would a seedless watermelon exist without humans? How did it become seedless in the first place? This thing has 3 sets of chromosomes. It's a triploid. Therefore, it doesn't set seed. It didn't exist before humans, and it couldn't exist without them. The same is true for navel oranges, also a triploid, also reproduced by vegetative propagation, like the way your grandmother or somebody might propagate African violets, or like apples are propagated by stem cuttings, or now often grapes. But the navel orange, seedless. Bananas, also triploid and seedless. These are also human inventions. The guy that developed the seedless banana was working, I think, for Chiquita, and had worked for 20 years to try to get this. Have you ever seen a wild banana? You can barely eat it. Its seeds are about the size of a pea, and there's barely any. You're spitting out more seed than you are eating banana, I'll tell you that. So if you look inside of this banana that we all take for granted, at 57 cents a pound, I might add, how does it grow for 57 cents a pound in Costa Rica, get picked, harvested, shipped to your grocery store, all for 57 cents a pound? But when you eat this thing, you see those black little dots in the middle? Those are seeds, aborted seeds that won't grow. So the banana is all vegetatively propagated. Seedless anything. But these giant tomatoes, where did these varieties come from? All of the different varieties now that we have of tomatoes. Have you ever seen a wild tomato? It's small and would bounce off the wall. We have now genetically selected for everything from aroma to a Burpee's Big Boy. And I love the Burpee's Big Boy. I'm so glad that somebody did that. All of these, then, human inventions for different qualities that people appreciate. This corn, I took this picture out of the Park Seed catalog, and this was produced by conventional breeding. This is not computer enhanced. Is this natural? Well, I just want all natural food, somebody might say. Really? I don't know what you're eating then. There are some plants that you can get in your grocery store that do exist in the wild, as wild plants. So you can go where the wild things are. You know, these are some examples, you know. And you can go to certain ethnic grocery stores and still get plants, cassava, and things that we don't eat here in this country very often. Some of those plants are also becoming domesticated. You can also find things now in our grocery store like these fiddleheads, the croziers of ferns, and these grow in the wild. You should be also a little bit aware that ferns are hyperaccumulators of heavy metals, so you might not want to pick these by your roadside, and be aware of where they do come from. They'll accumulate arsenic as well, but you know, so that brings up a whole nother question about how do we produce the foods that we eat? Where do we get them from? How do we treat them? This brings me to corn. The coming of corn has been a subject of intense scientific scrutiny as well as capturing the imagination and mythologies of Native Americans. There are a number of really interesting stories about corn and its importance to Native American cultures. There's a book by David Bruchac on various different Native American stories, and this one titled "The Coming of Corn" was from the Cherokee in North Carolina, about a grandmother who provided corn when hunting and game was scarce, and when her grandson saw this, she had to die, and the grandson dragged her body across the ground, and wherever her blood fell, a corn plant came up. And this was then how the coming of corn was mythologized in that Cherokee story. And corn did occur very suddenly in North America. It, unlike the domestication of rice, which may have occurred over hundreds, if not thousands of years, for the selection of rice that would stay on an inflorescence past harvest, all simultaneously, so it could be harvested at once, corn apparently appears very suddenly from its precursor grass known as teosinte. So the sudden domestication of corn then stimulated a lot of very interesting stories and a lot of interesting science. And John Dobley, who had been at the University of Wisconsin, has done a lot to investigate the mutations involved in the development of modern corn from its precursor wild plant, teosinte. And that looking at especially the contribution of two genes, one called teosinte branching and the other one called the gloom architecture gene, and how they influenced the domestication of modern day corn. So you see in this drawing off to the right, a drawing of teosinte, which is a branching type grass. It might grow 6 feet or so. And it has terminal inflorescence, and each grain, each fruit is surrounded by its own husk, in which hunting and gathering peoples would peel and eat, and typically there were about 8 grains per inflorescence, and both male and female parts of the flower were in that same inflorescence. So it were very small genetic changes which caused modern day maize to become monoecious, in that the male now is at the top of the plant that produces the pollen that we call the tassel. The female, which we call the ear, is arranged laterally. And it is by that separation that allows us to do controlled genetics on maize. And that we can take the pollen from one plant and self that ear. And that process can result in the development of inbred lines. And if you continuously self a plant like maize, you develop more homozygosity in the genes. And eventually, you can make an inbred line that is mostly homozygotic. And it was discovered actually by Darwin that if you cross two inbred lines, you result in a heterozygotic effect called hybrid vigor, that the resulting cross of two inbreds results in a hybrid vigorous response, resulting in higher yields if performed on maize. And this was first discovered here in the Northeast. So hybrid maize was actually developed in the Northeast, not in the Midwest, where one might presume. But it was George Shull, working out at the end of Long Island in Cold Spring Harbor Laboratories in the early 1900s, and Ed East, working at the Connecticut Ag Experiment Station down near New Haven, that first made inbred lines of maize by selfing them continuously, taking the pollen and selfing an ear, taking that progeny and selfing that, and selfing it repetitively through generations. The stature of the plants would grow less and less and less until they had developed line A in one direction and another one, line B. And then when they crossed line A and line B, bam, a big plant with a big ear. And so they developed hybrid plants in the early 1900s. And what this meant was higher yields. We measure yield in maize by how many bushels per acre are generated. How many bushels of ears can you get out of 1 acre of land? And before they were doing this, 50 bushels per acre would have been doing really good on your best variety. Say a flint corn that was grown around here in the late 1600s, or certainly by the Native Americans prior to that. But this process of making inbreds and then making hybrids created this increase in yield, and both George Shull and Ed East thought they were really into something and thought they could make a company out of selling hybrid seed to farmers because it would increase the yield. So if you could get 50 acres out of just the same old stuff that you normally grew, but you could get the seed from growing hybrids, and then all of a sudden you're making 70 bushels per acre, you're getting more money out of every acre that you grow. Good deal for the farmer. Now, if you were to take that seed that was hybrid and save it and plant it out next year, the genetics then start to fall apart again, and the yield then again drops. So this would become a business proposition. You could continually go back to these guys who would sell you their hybrid seed and get more bushels per acre, and it was worth the price of buying it. And this is a picture of George Shull's laboratory in 1906 at Cold Spring Harbor. This same laboratory produced another famous maize geneticist named Barbara McClintock, who in the 1920s, looking at maize genetics, discovered that DNA was not simply like a bead, a bunch of beads on a string and static, but actually segments of DNA were capable of jumping and moving around and reinserting, and what we call today transposable elements. And she discovered this in the late 1920s, wrote papers about it in the 1930s and 1940s, and was pretty much dismissed by the scientific community, owing to the fact that, one, she was a woman, and two, people just didn't have any molecular evidence for what she was observing. So that combination of factors figured into that her work was not substantiated until years later, after molecular biology came about, and in the 1970s and 1980s proved that she was right all along. And she received the Nobel Prize for her work that she had done years earlier when she was then in her 80s, in I think 1984. But it's this same laboratory that George Shull worked on the first hybrid maize plants, where Barbara McClintock did her Nobel Prize winning research on maize genetics at Cold Spring Harbor. And Cold Spring Harbor has produced a number of other famous scientists and some who have resided over that laboratory for a number of years, including James Watson and Stephen Della Porta. So owing to the fact that the male and the female are in different parts of the plant means that hybrid seed could be developed because you can detassel, remove the male from one row and force the pollination to the other row and collect that hybrid seed. And if any of you have ever grown up in the Midwest, this is a common job for high school students, to detassel corn. It's still manually done. They go into the field before the maize is pollinated, and they detassel that corn and suffer chigger bites and horrible weather and scratches by maize and all of this. The problem that Ed East and George Shull's technology had was this single cross that they did, A by B, was expensive to do. The seed production costs were too high to really make a company viable. And the inbreds were spindly and too weak and suffered from a lot of diseases. And the seed prices were just too high for the farmer to really consider it. Then along comes Henry Wallace in the 1930s. And what Henry Wallace discovered is he could make line A by line B and make the giant ear shown in this picture off to the left. And he could make a separate other two lines, we'll call them C and D, and again make a giant ear. And then when he combined those progeny in a four-way cross, he retains much of the yield increase to support the production method of hybrid corn. So it was the introduction by Henry Wallace for the production of the dihybrid cross that made hybrid maize commercially viable in the United States. And as soon as this was done, pretty much maize, the farmers adapted and adopted hybrids to replace open pollinated varieties almost overnight. In production areas like in Iowa, shown here, virtually all open pollinated maize disappeared within a decade. And it was all hybrid maize generated by Henry Wallace's company, which became Pioneer Hybrid, which controls a substantial market share of maize production today, owned by DuPont. There are other companies such as Monsanto, which have considerable market share in producing hybrid maize today. The reason why I'm dwelling on this is all maize today is hybrid maize. So it's all developed by human intervention using genetics. This is not genetically modified corn, this is hybrid corn. And I also bring this up because the farmer has to purchase this corn every year. Because again, the genetics fall apart if the farmer were to brown bag that seed. So there's a technology that is useful for the farmer because he gets more out of his land and hence more earnings per year out of the technology that the company provides to make hybrid corn. This sets the stage, you see, for using modern genetics to produce plants that are more useful by humans. I haven't even gotten to using advanced genetics techniques yet, or gene cloning techniques. I just mean to point out that all of these plants have been produced by humans and for the benefit of humans in agriculture. So now, if the average yield per acre before hybrid corn was developed was 50 bushels per acre, you'd be embarrassed to show up in Iowa now with a yield that is under 180 bushels per acre. Some boasting up over 220 bushels per acre. So we've increased the yield per acre 4 times by using genetics. So remember, the world is 7 billion people. It's going to be 9 billion people. How much arable land is left? If you can increase the yield per acre, this is a very valuable characteristic in breeding. No matter how you do that, you're getting more yield per acre by these technologies, and I haven't even gotten to genetic modification yet. Is it natural? Well, you know, it's been said that the man who has bread has many problems. The man who has no bread has one. So, biotechnology is not a threat. Starvation is. And when you talk to a plant breeder about the most important characteristics for plant breeding, the 3 most important characteristics in order are yield, yield, and yield, in that order. Anything that diminishes yield and takes a yield hit is not going to be acceptable for most crops. Because that's what the farmer depends on, after all. Unless it's replaced by some other quality that the farmer can be paid for and compensated for. Okay, so that concludes the first part on agricultural biotechnology. Where does our food come from? Not only where does our food come from the wild, and how we domesticated and selected varieties that end up on our grocery store shelves, but how we adapted to those plants and how we utilize those plants for large-scale agriculture. So in Michael Pollan's book, Omnivore's Dilemma, he examines large-scale agriculture, whether this is done organically or whether this is done in conventional agriculture. How do these large-scale techniques bring our food and the processing to our grocery store shelves? And I think the point that he brings up is really interesting, that whenever this process gets large, it gets complicated. He talks about the agricultural industrial complex, that certainly the organic industrial complex has its techniques and its motives also. The large-scale conventional agricultural complex has its techniques and its motivations. But certainly, how do we produce the volume of food that we produce? How do we utilize the techniques that are large-scale agriculture now to bring these products to market? And all of these could be brought into question and scrutiny. Is this the best way to do this? Is this the safest way? Is this the most environmentally sound way to do this? All of these things should be questioned. But for now, we've examined how our food gets to our grocery store via the selection from wild plants. We can take on these aspects of processing and production and some of the controversies it brings up later. In the next part, we will consider how DNA-based technologies affect food production.